Hi, everyone. Today we're going to be talking about DNA or deoxyribonucleic acid. Okay, so a couple things about DNA. Fun fact, maybe tell your mom today at dinner is there's 6 feet of it in every cell. Okay, so every single cell in your body, from your skin cells to your liver cells, the cells in your eye, et cetera, has the same DNA. Okay, they all contain 6 feet. Okay, what makes it unique is what genes are turned on. We'll talk about genes here in a little bit. DNA is composed of nucleotides. We'll talk about the structure of DNA here in just a second. But if you remember from our macromolecule unit, nucleotides have 3 components to it. Okay, there are 2 functions of DNA. Right, it controls protein production or enzymes. Okay, and then it can duplicate itself for new cells. All right, that's the main kind of jobs DNA plays in our body. The other thing it does is it can mutate, it can change, right, which can lead to new features or novel features in our bodies, which we talk about in evolution a little bit. A couple other vocab words: we have chromosomes and chromatin. Okay, we use DNA kind of as a generic term. We use chromosomes almost as a generic term as well, but technically they are chromosomes. You can see when they're in this X and Y formation, right? So this up there, right? You see that little X is our chromosome. Okay, if it's unwound DNA, so kind of near the bottom there, it would be called chromatin. Okay, we have 46 chromosomes in our body, right? And we'll talk more about that another day as well. So if we look at our DNA, right, each section of DNA is called a gene. Okay, a gene is a section of DNA that codes for a protein. Okay, and we have thousands of different proteins that we'll be needing kind of throughout our lives, and our DNA codes for that. Okay, so that's kind of how that works. So if you kind of look at DNA and how it's arranged, right, it's made up of nucleotides, right? Those are all put together to make larger strands of DNA, which are genes, and then you have a bunch of genes, or even just a bunch of nucleotides, kind of in a row to make then our chromosomes. All right, let's do that. Every human body, again, has 46 chromosomes, okay, except for your egg and sperm. We talk about that more in meiosis. You get 23 from mom, you get 23 for dad, for a total of 46 if you're good with math there, okay? Does this mean you have 46 genes? No, right? Again, a segment of DNA is a gene, okay? So you have hundreds of segments on each DNA piece, which means you have hundreds and thousands of genes, okay? Here we go. Let's get to the nitty gritty structure of DNA. DNA, right, we said is made up of a nucleotide. Nucleotides have a base, right, which can be an adenine, thymine, cytosine, or guanine. So that's our rectangle there. It has a sugar, which in this case is a deoxyribose sugar, usually drawn by a nice cool pentagon. And it has a phosphate group, which is usually drawn by a circle there, okay? So you can see that circle represents our phosphate. So again, general structure here, again, base, cool square or rectangle, sugar, pentagon, and then this circle thing would be our phosphate, okay? The bases can be 4 different things, as I mentioned. It could be adenine, thymine, cytosine, or guanine. Generally, we just write A, T, C, or G. Makes it a lot easier, okay? If we look at these bases, right, we see that adenine and guanine, right, so adenine here, guanine here, are both double ring structures, and they're called purines. Okay, cytosine and thymine down here are single rings, and we call those pyrimidines, okay? And that's how we find these bases, okay? And again, thinking that phosphate, the sugar, terrible drawing, and then one of these, an A, a T, a C, or a G, will be connected to that to make each then nucleotide 4 different nucleotides. This dude, Chargaff, right, came around one time. They were studying DNA, and he basically started breaking apart DNA and was asking questions about it. And he found that the amount of guanine in a DNA strand is about the amount of cytosine in a DNA strand. Same thing with adenine. Adenine, right? The amount that there is of adenine is going to equal the amount of thymine. So from that, we get Chargaff's rule, where wherever you see a C, you have an equal G. When you ever see an A, you have a T. So if your strand of DNA lines up like A, T, C, G, right? This A is going to pair up with a T. The T is going to pair up with an A. C pairs up with a G, and therefore G pairs up with a C. Okay, so that's kind of how that works out. We can see down here in this chart too that it works out kind of through all species. It's not just mammals or humans or anything like that. It kind of works out through everything. Some other people who are really instrumental in discovering like the structure of DNA, Rosalind Franklin gets no credit for it practically at all because unfortunately it was kind of like a man's world, and so there's this race to figure out structure of DNA. She kind of helped with it, but didn't really get any credit. Okay, she's infamous for taking photo 51, which is right here. Okay, X-ray diffraction kind of showed that DNA was a double helix. Okay, so you can kind of see these lines, right? And from that, she was able to determine DNA was a double helix. Well, unfortunately, these guys, Watson and Crick, right? So these guys right there, Watson and Crick, basically stole her information and created the structure first, right? So they created this model that DNA is a double helix, okay? They likened it to a twisted ladder, okay? So it's not this straight line, right? So it doesn't exist like that. It's actually twisted, okay? On the outside, it goes sugar phosphate. Okay, so it alternates. So the back would be like sugar phosphate, sugar. If I can draw this phosphate, and the middle is the base. I'm going to try B for base. Right, it just keeps alternating like that. Okay, and again, those bases could be an adenine, guanine, thymine, or cytosine, and it's double-stranded. Right, so it means on this side, it's not just a single strand, but this base pairs up with another base, and then there's the sugar that kind of connects them, and the phosphate, and so on and so forth. Okay, in between, what connects those bases here is a hydrogen bond. Okay, between the adenines and thymines, there are 2 hydrogen bonds. Between the guanines and cytosines, there are actually 3 hydrogen bonds holding that double helix together. Here is their model, right? You can kind of see, again, outside sugar phosphate, inside the base, held together by hydrogen bonds. A couple other things about DNA. We know DNA is complementary, which just means that the bases on one strand match the bases on the other. So, for example, if we have this strand here, this is strand 1, strand 2, right? This A would match with the T would match with an A, G would match with the C, this C would match with the G, and so on and so forth, kind of all the way down. And that's what you would have. So you have one strand being 1 and the other kind of being the opposite there, based on Chargat's rule. Okay, and there it is. I didn't even have to write it. At least know I was right. The orientation of DNA, right? So if you number carbons, you get like 1, 2, 3, 4, 5. And so we have like a 5' end, we call it, and we have a 3' end. Okay, that's what we call the orientation of DNA. So on one side is the 5', the other side is the 3'. Okay, so when it goes the 5 to 3' direction. So if we look at that DNA is complementary, if this is the 5' end, we know that it'll match then to the 3' end, and this 3' will match to the 5' end. Okay, looking then like that DNA strand. Right, so one goes one way, and the other strand goes the other way. Okay, so it's a complementary matching, and the strands go the opposite directions. Kind of look at DNA through prokaryotic and eukaryotic. We talked about this a little bit in our cell unit, but prokaryotic DNA, remember, is very simple. Okay, it's circular, kind of hangs out in the nucleoid region. Okay, whereas eukaryotic DNA, when we talk about DNA, we're really talking about eukaryotic DNA. Okay, we have the chromosomes; they're linear, and there's a whole bunch of them, right? 46. Cool thing is plasmids. Okay, prokaryotes, bacteria specifically have plasmids, and plasmids are just basically extra DNA that will provide maybe antibiotic resistance or something like that, some other genetic advantage. All right, and that's all I have for you. So hope you enjoyed that.